Welcome to yet another session of zoology. Today's topic will be dealing with the structure and types of chromosomes. Let me introduce myself. This is P. Karnakumari, Assistant Professor of Zoology, currently working at Sangareddy. The structural types of chromosomes is a part of cell biology, which is a part of fourth SEM syllabus for the second year students. Let us proceed further. So in today's session, we'll be talking about what a chromosome is and what are the structures and functions of the chromosomes. So basically what a chromosome is a DNA, or it's a deoxyribonucleic acid molecule that has got a part or all of the genetic material that is genome of an organism. Now, this was first identified in a eukaryotic cell, and it was demonstrated by E. Starsberger in the year 1875. The term chromosome was given by W. Walder in the year 1888. The word chromosome is derived from the Greek origin, where chroma means color and soma means body. Now, this is because this particular chromosome has got the affinity towards the basic dyes when they take up when you see it under the microscope. Now, chromosomes are generally not visible in the interface or in a metabolically active nucleus, as they are in the form of a network called chromatin network. During the cell division, the chromatin network undergoes spiralization and condensation and becomes progressively thicker so that they can be easily separated during the cell division. So in eukaryotic cells, as we can see here, DNA is tightly packed into a thread-like structure called as chromosome. So this is the DNA, which is double helix structure. And when the cell is undergoing the cell division in the metaphase, you come across the DNA in the form of the chromosome here. So it is now with reference to the prokaryotic cells. This is in contrast to the bacteria, where DNA it floats freely around the cells. Now, a single length of DNA, as you can see here, is wrapped around proteins called histones to form a structure called nucleosome. So this is the DNA that is coiled tightly around the histones to form nucleosomes. And nucleosomes as are further coiled up tightly to create a chromatin loop. And this is far more uncondensed chromatin fiber when compared to the metaphase chromosome. Now, this uncondensed chromatin fiber, which is formed by the condensed nucleosomes, further undergoes condensation to form loosely condensed chromosomes. Here, these are the loops of the chromatin material. Now, these are subjected to supercoagulation, condensation of the chromatin, which finally is revealed as condensed chromosomes during the cell division. Now, the chromatin loops are then wrapped around each other to make a full chromosome. The DNA making up of each chromosome contains thousands of genes. At the end of each chromosome, here, basically on the both top and the bottom, are sections of DNA called telomeres. Telomeres protect the end of the chromosome by forming a cap. They form a cap, a protective cap at the ends of the chromosomes. Change in the number of chromosomes, that is referred to as aneuploidy or structure, which means chromosomal mutations of chromosome. It causes disorders. Now, a particular chromosome consists of the following structures. To start with, the very first one is called a centromere, or you can also call it as a primary constriction. As you can see in the image, this is the primary constriction, otherwise also called a centromere. Now, this is a lighter staining region that appears as a constriction. Spindle fibers are attached here during the cell division. Now, apart from the primary constriction, a chromosome also has got a secondary constriction. There can be one or more may be present on a single chromosome. As you can see in this image, this is one particular secondary constriction that we are coming across in the image, and then yet another secondary constriction in the same image. So it is possible that there can be one or more than one secondary constrictions present in a chromosome. Now, these secondary constrictions uh, it is often associated with the nucleolus. This particular part is often associated with the nucleolus during interphase and takes part in the reorganization of nucleolus after cell division. Hence, it is called nucleolar organizing region after cell division. At this region, genes for rRNA synthesis are present. The next part or the next structure associated with the chromosomes are the satellites. You call it as satellite body. The part of the chromosome above the secondary constriction. So this particular part above the secondary constriction, which looks like a bulb. This is present here, which looks like a bulb, as you can see it here. Which is present at the end of the chromosome beyond the secondary constriction, and it is called as satellite body, and the chromosomes which bear the structures are called as sac chromosomes. So the chromosomes in humans, that is 13th, 14th, 15th, 21st and 22nd chromosomes, contain a segment that is separated from the main body of the chromosome by such a secondary constriction. So all these 13, 14, 15, 21 and 22nd chromosomes of humans are referred to as sac chromosomes. The secondary constrictions are always constant in their position and hence can be used as markers to identify these particular chromosomes. So this is about the satellites. Then coming to the following parts of the chromosomes, we have what are called as arms. Now, a chromosome contains two arms divided at the centromere, as you can see it here. There is a short arm that is referred to as P arm, and there is a long arm that is referred to as a Q arm. Then what is the telomere? We have already discussed. The ends of the chromosomes which are capped by DNA and that protect the chromosome are called as telomeres. Then kinetochore, as you can see in this image, this is the kinetochore, is a disc-shaped protein structure that is found at the centromeric region of the chromatid, to which the microtubules of the mitosis or cell division are attached during the cell division to this particular structure. Then coming to chromatid, the yet another part of the chromosome. Each chromosome, as you can see, the whole thing is a chromosome. This is made up of two identical parts called as chromatids. And two chromatids are linked at the centromere. So this is a sister chromatid, and this is a sister chromatid with respect to each other. So these are the parts of a chromosome. So a chromosome on a whole consists of sister chromatids that are attached or linked to each other at the centromere. So this is one chromosome we are talking about, and this is the one chromatid. And when copying of DNA material takes place and chromosome condensation takes place, one chromosome replicates another chromatid. So this is where you have two sister chromatids. Both are held together by the centromere. Now, during the cell division, chromosome separation takes place, and that is when there is a split in the centromere, and the two chromatids, the sister chromatids, are separated from each other. The sister chromatids are separated or grouped on each other, and they will be representing the two chromosomes, which will be going to two different daughter cells at the end of the cell division. Now the chromatin fiber, what is actually present here, it would undergo condensation to form chromosomes. So during interphase of each chromosome, decondenses to form the chromatin network at the end. The other parts of the chromosome consist of pellicle and matrix. Each chromosome is bounded by a membrane called pellicle, and this membrane encloses a jelly-like substance which is usually called matrix. So in the matrix is present, the chromatonema is present. The matrix is also formed from achromatic or non-genetic material. So let's go back and just check. So this is the pellicle or the outermost covering of a chromosome, and within this you find a jelly-like substance, and inside the jelly-like substance we come across the chromonemata, that is the chromosome material. So we have seen what a primary construction is, we have seen the secondary constructions, we have seen the satellite bodies, then we have discussed about the pellicle and the matrix. To continue further, let us talk about the size of the chromosomes. The size of the mitotic chromosomes of various plants and animals varies from 0.5 microns to 32 microns in length and zero point 0.2 microns to 3 microns in diameter. The giant chromosome found in the cells of salivary glands of diphtheria is permanently in pre-metaphase stage and is visible in the interphase nucleus. These chromosomes are approximately 300 microns in length and 10 microns in diameter, but otherwise generally the plant chromosomes are longer than the animal ones at any given time. So chromosome number is a precise number. It is typical to of a given species. So any species, any member of a one particular species would be always having a fixed number of chromosomes. As in our case, for example, humans have got a total of 46 chromosomes. That is 23 pairs of chromosomes, and therefore it's going to remain same for all the human individuals. Now, in any given asexual reproducing species, the chromosome number is always the same, asexual. And in sexually reproducing organisms, the number of chromosomes in the body cells, that is somatic cells, is typically is always diploid into one state, that is a pair of each chromosome, and twice the haploid, that is one n number found in the sex cells or the gametes. So in the sexually reproducing organisms, the number of chromosomes is half. Different species have different number of chromosomes. We have seen this, humans are diploid. Now these 46 chromosomes was organized into 23 pairs, where 22 pairs are autosomes and one pair are allosomes, that is sex chromosomes, which help in determination of the sex of the humans. Now, based on the type of their centromere, there is further classification of the chromosomes, as we can see. Now, we all know that the number of chromosomes serve as an aid in determination of the phylogenetic status, such as taxonomic position of plant and animal species. In higher animals, each somatic cell contains one set of chromosomes inherited from a mother and a comparable set of chromosomes inherited from the father. So now coming to the classification of the chromosomes, depending on the position of the centromere, chromosomes are classified into four categories, that is metacentric, submetacentric, acrocentric, and telocentric. Once again, it is metacentric, submetacentric, acrocentric, and telocentric. In metacentric chromosomes, the centromeres occurs exactly in the middle or at the center. That is, two arms of the chromosomes are equal. The P arm and the Q arm are equal of such chromosomes. And the metacentric chromosomes look as V-shaped during anaphase movement. So this kind of chromosomes are called metacentric chromosomes. In the submetacentric chromosomes, centromere is located to one side of the center point. That is, centromere is submedian, not in the center, that is between middle and the end. so that it divides the centromere into two unequal arms, a short arm and a long arm, and they look like L shape in the anaphase. So this is L shaped in the anaphase. This is the long arm and this is the short arm because the centromere is somewhere between the middle and the end. In acrocentric, or you can also call this as subtelocentric or subterminal centromere, the centromere is situated almost near one end or close to an end of the chromosome. That is, the centromere is subterminal in position and it gives two arms, one exceptionally short arm and the other a long arm. Now these acrocentric centromeres, they look like J. They look like J during the anaphase. Usually the locust and some grasshoppers are acrocentric. Now the last type of the chromosome is called as centromeres appears to be located at one end of the chromosome, that is in the position which is normally occupied by one of the telomeres. So in this type, one arm is more or less equal to the length of the chromosome and the other arm is beyond the centromere and is represented simply by a dot. So here you find that the centromere is present at the end. Now, telocentric chromosomes are of very rare occurrence. Telocentric chromosomes exist normally in certain species of homomastigote protozoans. Usually each chromosome has only one centromere, but in some species, each chromosome has more than one centromere. Again, in some cases, the centromere is absent. So these are metacentric, exactly in the middle, submetacentric, somewhere between the middle and the end, acrocentric, that is close to the end, and telocentric is always at the end. So this is the classification of chromosomes based on the centromeres. So once again, this is the metacentric, exactly in the center with the both arms. Submetacentric with a short arm and a long arm. Acrocentric, this is always close to the centromere with a very short arm and a long arm. Telocentric, because this you cannot find it in humans, where it is almost present at the area where you accept the telomeres to be present. Now moving further, let us talk about karyotype, what a karyotype is. Karyotype is an organized profile of chromosomes in which the chromosomes are arranged according to the size and position of the centromere and numbered from the largest to the smallest. So the profile of organism chromosomes, which are organized by their size pattern and location of centromeres and numbered from the largest to the smallest. Now, a karyotype is the characteristic chromosome complement of a eukaryote species. The basic number of chromosomes in the somatic cell of any individual or a species is called the somatic number and it is designated as 2n. We know that. And in normal diploid organisms, autosomal chromosomes are present in two copies and sex chromosomes may XX or may not be like it can be XY to be present in pairs. So according to international conventions, human autosomes are numbered from 1 to 22 in decreasing order by size, and with the exceptions of chromosomes 21 and 22, which are the smallest autosomes, the sex chromosomes are generally placed at the end of what is called as a karyogram. So a karyogram is where the chromosomes are aligned along a horizontal axis shared by their centromeres. So individual chromosomes are always depicted with their short P arms, P for petite, that means small at the top, and the long arms, Q for Q, at the bottom. So centromere placement can always be used to identify the gross morphology and shape of these particular chromosomes. So this is the human karyotype of a male, and this is also the karyogram of a human male, where we can see the chromosomes are arranged starting from the largest chromosomes to the smallest towards the end. So these chromosomes, as you can see, are further classified, based on their centromeres. They are further divided into 7 groups and sex chromosomes, as you can see, A, B, C, D, E, F, G, and this is the sex group. So coming to the first group, now these are large metacentric chromosomes, as you can see. Now this is the line where the centromeres are being aligned. So first one is large metacentric. Then you have B group. This is large submetacentric chromosomes. Then we have a C group that includes from 6th, 7th, 8th, 9th, 10th, 11th, and 12th chromosomes. These are medium-sized submetacentric chromosomes. Then we have next group, that is D group, that includes 13, 14, and 15 chromosomes. These are medium-sized acrocentric chromosomes, and that includes satellite bodies as well. Now, E group consists of chromosomes numbering from 16, 8, 17, and 18, and these are short metacentric chromosomes. Now, 19 and 20 are also short metacentric chromosomes. Coming to the G group, that is 21, 22, and Y chromosome here, as you can see, are with the satellites, and Y chromosome doesn't have a satellite. So this is a classification of chromosomes that is based on Danier, and these are the groups into which the chromosomes are classified as A, B, C, D, E, F, G. Now, this is what we have discussed. Now, chromosomes from 1 to 3 are large and metacentric. Chromosomes from 4 to 5 are large and they're submetacentric with relative position of centromere. Chromosomes 6 to 12, including the X chromosome of the sex chromosomes, are medium in size, and so they are submetacentric. Group D consists of chromosomes from 13 to 15. The size is medium, and with relative position of centromere, they are acrocentric. Group E is 16 to 18. These are relatively short chromosomes, so they are submetacentric with the position of centromere. Group F, that includes 19 and 20, are very, very short chromosomes. They are metacentric or submetacentric. And the last group, that is G, it includes 21 and 22 chromosomes, including Y chromosome, and this is short and acrocentric. So this is the classification of chromosomes based on their centromeres. I hope this session was of some use to you. Thank you.